Thank you for being here. And my talk might be a little bit surprise for those of you who think that climate change is only a bad thing, because today I'm going to give you an example where actually seagrasses might be winners under changing climate conditions. First of all, seagrasses are keystone species because they provide habitat for many different organisms. So you can think of them as like the forests that are existing underwater. So similar to terrestrial plants, they do produce oxygen, but they do it underwater. And they do this through photosynthesis. As all photosynthetic organisms, they also do respiration. So if we think photosynthesis as an income where they can accumulate carbon into a sugar pool, respiration becomes like an expense where they're using some of this carbon from this sugar pool. The balance between photosynthesis and respiration is very important for the plants so that they can keep some of this sugar pool for a long-term balance, which we can think about as a savings account. So if there's enough carbon reserve in this box, that means they can grow. And indeed, if there's enough of it, they can make further investments where they can produce more plants and more shoots, and which we can measure as the survival. All these balances are regulated by environmental factors. The two important factors that regulate photosynthesis is the available light and carbon dioxide, which I'm going to say CO2 from now on. So they need both of these parameters. If either of them is limiting, their photosynthesis is not sufficient enough for the plant to keep up with the sugar pool. So, for example, under limiting CO2 conditions, which are in today's ocean conditions, the available light becomes excessive. So what that means for the plant is they use the light with photosynthetic pigments, but if the light is become excessive, they have to protect their photosynthetic pigments. For that purpose, they use photoprotective pigments, which you can think about as a sunscreen. So in summer, you know, during hot summer days when we have a lot of sun, we put like sunscreen on our skin so that our skin doesn't get burned. So the plants are using a similar mechanism. They use this photoprotective pigments not for their photosynthetic pigments not to get burned. The third parameter that is important is the temperature. However, temperature affects respiration rates more drastically. Eelgrass usually prefers to live in cool environments because increasing respiration rates, increasing temperature, increase their respiration rate so much so they cannot accumulate enough carbon in their reserve, which translates into decreased survival. So that's why the distribution of eelgrass is limited to temperate regions. Indeed, the eelgrass distribution on the east coast of the United States and around North Carolina boundary. So ideally, seagrasses need adequate amount of light and CO2 for their photosynthesis to keep up the sugar pool and favorable temperature for their survival. But globally, we know they are stressed due to the increasing temperature because of the global warming. We have been experiencing periodic dieback events in Chesapeake Bay due to high water temperatures in summer months. We also know that CO2 concentrations in today's oceans is not enough for their photosynthesis. Because CO2 is limiting their photosynthesis, the available light becomes excess, which creates the need for a photoprotection mechanism. So if everything is linked to CO2, so how is increasing atmospheric CO2 concentrations are going to affect seagrasses? First of all, the CO2 is not only increasing in atmosphere. Whatever is increasing in the atmosphere is taken up also by the ocean, which we term as ocean acidification, and this is changing the pH of the seawater. So if the CO2 continues to increase in the seawater, can this decrease the damage by the excess light? So in that case, what is going to happen to photoprotection? Can increasing CO2 also decrease the stress effect of high temperatures because they can do more photosynthesis and accumulate enough sugar? More importantly, why do we care to quantify all these interactions? Because based on these interactions, we can make predictions, and which will allow us to give advice for the restoration projects of eelgrass in the Chesapeake Bay region. So, and for all of these, to test all of these hypotheses and many other questions, which I don't have enough time in this talk, we have been running a climate change experimental facility at Virginia Aquarium. We have 20 tanks at 5 different CO2 treatments, and each tank we have 5 seagrass strays which were collected from Eastern Shore in May 2013 and transplanted in May 2013. So we are and the maintenance of this system requires a lot of effort. by many people. So we have been collecting monthly plans to measure the growth rates and survival, as well as tissue carbon, nitrogen, and pigment content. We also use plants for laboratory measurements. We are continuously monitoring and recording the temperature, the salinity, the light, and also the pH as a proxy of the CO2 concentrations. We change the CO2 concentration by bubbling CO2 gas from the gas tank. And CO2 tank. And before I move forward to the results of the first year, I would like to mention this is still an ongoing project, so you are all welcome to visit us at the Virginia Aquarium to see this facility, and you can even volunteer maybe. So, and I would like to say, like, this is a very quick snapshot of the results, so if you have further questions, you can find me in the poster session. So, after 4 months of giving them CO2, we could easily see a difference between among the treatments. When you give them high CO2, the plants were much bigger. And these graphs are showing how the differentiation happened along the time. On both of the graphs, I have on the x-axis the time, so from May until November, and on the other y-axis, I have the CO2 concentrations. So in May, they all started at the same conditions because they came from the same population. In June, we turn on the CO2, and throughout the summer, the high CO2 treatments not only get bigger, they also got fat. We could also see a big difference between their growth and survival. So if they had higher CO2 concentrations, we had bigger plants, as I showed before, and their numbers were also higher. And these graphs are showing when the differentiation in the growth and survival started. So the differentiation in the growth rate started after the summer. In summer months, the seawater temperatures reach 30 degrees Celsius. So this is beyond the heat threshold for eelgrass, so which puts them into the stress where they cannot accumulate carbon reserves if they don't have enough CO2. But giving them high CO2 allow them to accumulate the carbon even under high temperature stress conditions, and where we had the higher survival rates at high CO2 treatments. Opposite to the increased biomass and the leaf size, we saw a decrease in the pigment content. These graphs are contour plots in which are showing the photosynthetic pigments, the concentrations of photosynthetic pigments and photoprotective pigments. So on the horizontal axis, we have the time, and on the y-axis, we have the CO2 concentration. And the darker the color, the more pigments there are. So if it goes from dark being pale, it means the concentration decreased. And as we can see throughout the time, for both of the type of pigments, the concentrations went down. But it was more drastic at high CO2 treatments. So it went down like almost half of it. By knowing the pigment concentrations and how much light is absorbed, taken up by the plant, we can calculate a parameter which is called optical cross-section. This parameter estimates how efficiently the photosynthetic pigments are capturing the light. And if you remember, I told you like they have less pigments, but as you can see, at high CO2 treatments, although they had less pigments, they were much more efficient capturing the light energy and to convert that energy into biomass. So this is a puzzling question, because this response is seen if they are living in a high light environment. But what we provided them is more CO2. So why they are giving this response to increasing CO2 similar to it being high light is one of my questions. I'm working on it. To conclude, ocean acidification increases carbon accumulation of eelgrass shoots by increased photosynthesis, and therefore their survival even stressful conditions, for example, at high temperature stress environment. In addition to my academic research support, a Virginia Seagrant Fellowship provided me the opportunity to collaborate with Virginia Aquarium staff to promote ocean science education. I have participated in many different education programs which taught me how to teach science in a daily language, and I'm still working on it. And with that, I would like to thank to my advisor, Dr. Zimmerman and Dr. Hill, and my committee members. It will be impossible to run all these experiments without all the effort from the Biooptical Research Group members. I'm also thankful to my outreach mentors from Virginia Aquarium to teach how to communicate science. And I'm grateful to Virginia Seagrant both for funding my research and also giving me advancing my academic skills. Thank you.
We have time for some questions.
I have a question. So the range of pH values that you ended up with, are those reasonable in terms of projections for ocean acidification in the next 50 or so years? Or it seems like 6 was a little bit low.
Yeah, it is quite low.
Maybe
So
7.
yes. So we have a couple ranges which are more realistic, but the lowest we are trying to estimate the physiological response. So this is not just replicating the environmental. If we are trying to see where the optimum is and how this relations if we quantify across a region, and then we will have the capability of to predicting. So it's not just a comparison low versus high. We are trying to quantify the relationship to incorporate into the models.
The change in absorption or the amount of chlorophyll in the cells and the efficiency suggests that it's being packed into the cells differently. Do you have any idea if the plant structure is changing so that it changes how it fits the chlorophyll?
Yes, actually there's more data which I haven't presented, so where we're looking at the package effect, so because depending on the light they can put more pigments and that will shade, that was what I was discussing. We saw an increase in the biomass per area, so that means the plant might be getting thicker or it might be getting more dense, so which will change the pigment orientation in the plants. We have some pictures with the microscope where we took to quantify the changes in the cell structure, but that is still an ongoing project, so I haven't had that data.
Any other questions? Great. Thank you very much, Billur.